Hi guys, and welcome to our final lecture. I have now lost the mop, so now we can focus on our last lecture and hopefully end the semester on a high note. So today we'll be talking about what is probably my favorite topic area that we have discussed during our lecture series, and the one that I'm probably the most familiar with, and that is aquatic microbiology. And we're also going to be talking a bit about water quality. So yeah, let's get cracking. So the first thing, obviously, is just to provide you guys here with a definition of aquatic microbiology, which is the study of microorganisms and activities in natural waters. So this, with inherent in this system, you have, or in this definition, you have to realize that this definition will apply to both oceanic and freshwater systems. Um, and typically, we do divide our water systems into two categories, lentic and lotic systems. So a lentic system is a system that has stagnant water. So water that is quote unquote not flowing or not moving. So think of something such as a lake or a pond. That would be classified as a lentic system. And on the other side of this, you have a lotic system. And this low sounds like flow. So as you can imagine, this would be flowing water. So think of things such as rivers or streams. I do want to note, however, we do use the term stagnant water, and we do use conceptually talk about water that is not moving, but it is worth knowing that all water is technically moving. It just takes way more time in certain instances than in others. So this division of water into these two systems is useful to do, but don't think that the water in lentic system is completely stagnant in that system, that there's no movement. Water will always move, or almost always move at least. So, and then something else that I do want you guys to note here is, if you can recall from our soil microbiology lectures, the microbe diversity and numbers in water systems are typically much less than in soil environments, so in terrestrial environments that you are used to. But there are exceptions to this, where large numbers of microbes can be found in water systems, and that's typically indicative of very high nutrient levels. So in instances such as eutrophication, where you have this massive influx of organic matter that these microbes can use to proliferate, then you will still then find very high numbers of microbes. And in these microbes, so let's talk about the microbes here for a second. You will note that you can divide the microbes also into two main categories. You can have pelagic microorganisms, which will be further divided into planktonic and nectonic. And then you can also have benthic microbes. So first, let's start with what's a pelagic microbe. So pelagic organisms are those that live in open water. So think of it as living in open ocean water that is not associated with the bottom. So these microbes are floating around in the water column itself. And then within pelagic organisms, you find planktonic organisms. So these are essentially a diverse collection of organisms that are found in water that are unable to propel themselves against the current. This actually constitutes the majority of microbes. So these microbes cannot move against the current. They can't swim against the current. So essentially, they are just floating around and moving as the currents move. In the ocean, these planktonic microbes are an absolutely crucial food source to many small and large aquatic organisms, including fish and whales. So it's really, they play a vital role despite their small size. Now, on the other hand, you have nectonic, and as you can probably infer at this point, these are aquatic organisms that are able to swim and move independently of water current. So they can actually move against the current, or they can, if there's perhaps a weak current or no current, these organisms can still move around. In terms of microbes, this would be in a far minority when compared to planktonic organisms. So just keep that in mind. Planktonic is by far in the majority between these two. Then we will move on to benthic organisms. So benthic organisms are organisms that are attached to sediment or to the surrounding walls of river or banks of rivers. So they are essentially where your pelagic organisms are freely floating. If this is your water body and you have your pelagic organisms floating around here, your benthic organisms are organisms that are attached like to the bottom or to the sides of whatever your water body is, be that the ocean floor or be it a riverbank. And they can also be attached to sediment. So typically these bacteria or microbes will have appendages and holdfasts so that they can actually attach to these surfaces where they will then form, where they will then grow and live. Okay, so moving on, we're going to just look here about the different habitats and how they determine which microbes can live where. So first, you should be aware of your littoral zone. So your littoral zone is essentially along the shore, as the notes state, and you will find rooted vegetation here. Then we will find the limnic zone. So your limnic zone is here, this upper, almost surface and just below the surface. And this is essentially where you have the surface of open water away from the shore. And here you will find many photosynthetic microbes, such as the algae. You can imagine this because they still are exposed to incoming sunlight at this depth. As you move a bit deeper, you will reach the profundal zone. So essentially here, this zone, as you can see depicted here, it lies beneath the limnetic zone. Here you will find lower oxygen because the limnetic zone is also in contact with the atmosphere. So as you continue downwards, you will have a decrease in both oxygen concentration and a decrease in light. So you can think about how that will impact your microbes. So here you will have lower oxygen. So there will be a predominance of anaerobic purple and green photosynthetic bacteria. Remember, those bacteria are capable of photosynthesis in the absence of oxygen. And then right at the bottom, here you find your benthic organisms. Remember, they are attached to your sediment, so they're the bottom sediment. Here you will find typically, if the depth is great enough, you can find no light or oxygen. So here you can find organisms that can potentially be methane producers, such as desulfur Vibrio. Something that I do want to point out on the sketch here, you might notice the term here at the top, oligotrophic. So essentially, an oligotrophic environment can be thought of as the inverse of a eutrophic environment. So that means that an oligotroph is an organism that can live in an environment that offers very, very low nutrient levels. The open ocean is a good example of an oligotrophic environment. These oligotrophs can also be contrasted with copiotrophs, which prefer nutritionally rich environment. You can obviously infer that from the name copiotrophs, like they need copious amounts of nutrients. Oligotrophs, you can also, I expect you guys to be able to infer this at this point, that because of their low abundance of food, oligotrophs are typically characterized by very slow growth, low rates of metabolism, and generally low population density. All of this should still make sense for you guys following from the metabolism lectures we did, so I do expect you to be able to realize that at this point. Okay, so now we come to a slide where there's also going to be a lot of information, because ideally I would have liked to split this lecture into two parts, doing one on freshwater and one on seawater, but we didn't have the opportunity to do that, just a bit of time constraints. So anyway, let's dive into seawater microbiota. So first, we're going to talk about phytoplankton, which essentially you will find in the top 100 meters of the ocean. So here you can see the depths. So in this top portion here, also known as the photic zone, you will find photosynthetic cyanobacteria that can fix carbon and nitrogen. Sorry, my neighbor with his Harley Davidson has just arrived home. Organisms that you would be able to identify in this area includes your Prochlorococcus, your Synecdoche, and your Trichodesmium. So important here, this Prochlorococcus is a genus of very small marine cyanobacteria with an unusual pigmentation. They have both chlorophyll A2 and B2. These bacteria belong to the photosynthetic phytoplankton and are probably the most abundant photosynthetic organism on the planet. These microbes are among the major are among the major primary producers in the ocean and are responsible for a large percentage of the photosynthetic production of oxygen. They are so abundant that a single milliliter of surface seawater may contain 100,000 cells or more. So there really are a bunch of these things. Then moving on to Synecdococcus. Synecdococcus, they are a unicellular cyanobacterium that is very widespread in the marine environment. The size varies between like 0.8 to 1.5 microns. They are essentially photosynthetic coccoid cells. You can see that from the coccus name, so they're spherical cells. And are preferentially found in well-lit surface waters where they can also be pretty abundant. Then moving on to Trichodesmium. These are also sometimes known as sea sawdust, and they're a genus of filamentous cyanobacteria that are found in nutrient-poor tropical and subtropical ocean waters. So then moving on to below 100 meters, here you will start finding that Archaea will dominate. Remember that Archaea are usually the organisms that are extremophiles, so they can dominate in these colder and high pressure environments that the lower ocean, those conditions in the lower ocean. The same can be said for the seafloor sediments. You also have Archaea dominate. With huge populations there, and then you will find chemoautotrophs here that will sometimes also live on these hydrothermal vents. I hope you guys can recall that we saw some pictures of them earlier in the course. So in the deep water, in the pelagic zone, there's a contrast between the water there and the water that is near the coast or on the continental shelf. And you will find a difference in your microbes that can occur there. For the primary difference being that you have this dominance of Archaea, and that's because they are able to inhabit these extreme environments, as I have said. Okay, so moving on, this is what our movement of organic matter will look like within the ocean. And so I'm just going to run through all these numbers, giving you a brief description of each. Please make sure you understand how this cycle works. It's important for the exams and the tests, or the upcoming exam, I should say. So first, here at number 1, you will see that you will see the predominance of phytoplankton. So these are those plant-like organisms, with phyto actually coming from the Greek word that means plant. They are autotrophs, so they generate their own food and they do not need to consume other organisms. You can see that as they take in CO2 and sunlight, and that is how they exist and how their metabolism functions. They actually perform 3 very, very, very important functions in the marine environment. Firstly, they generate nearly half of the world's atmospheric oxygen. Secondly, they regulate the ocean and atmospheric carbon dioxide levels. You can see with this exchange that happens here. And they also form the base of the marine food web. So these phytoplankton are absolutely crucial, not just in a marine context, but just in a global climate context also. Now we will move on to number 2. Here, this number 2 represents sinking particles. This can be divided into 2, either DOM or POM. So DOM is dissolved organic matter, and this is the fraction of organic carbon that is operationally defined as particles that can pass through a filter with a pore size between 0.2 and 0.7 micrometers, approximately. The definition might vary slightly depending on the source, but it's the point being it's the smallest particles. It's much smaller than POM, which we'll get to in a second. So various mechanisms are actually responsible for the DOM net accumulation in the ocean, such as phytoplankton release, grazer-mediated release and excretion, release via viral and bacterial cell lysis, particle remineralization, release from prokaryotes. So there are many, many processes that contribute to this accumulation or this increase in DOM in the ocean water. In the water, the DOM will then undergo many biotic and abiotic structural changes, resulting in the formation of various organic compounds. So therefore, DOM is a complex mixture of molecules that are produced by biotic and abiotic processes in the ocean. Very important. So now moving on to POM. This is particulate organic matter, and essentially this, you can think of these as similar particles to these, they're just much larger. So this is the fraction of total organic matter that is operationally defined as that which does not pass through a filter pore that typically ranges between the size of 0.05 and 2 millimeters. So you can already see that's a much different scale. It's much, much larger, these particles. And that's why these particles are considered non-dissolved and these are considered dissolved. So now moving on to number 3, so we're jumping back up here. This is essentially where we will find our zooplankton, and zoo, as you can imagine, or it comes from the Greek word meaning animal. Any of you guys studying or having studying animal sciences should be well aware of that. So these are your heterotrophic organisms. So as you guys know, they cannot produce their own food and must consume other plants or animals as food. So in particular here, this means they eat the phytoplankton. So to just simplify this for you guys, you can almost think in a terrestrial environments, these phytoplankton would be similar to plants, and then these zooplankton would constitute other animals that would then feed on the plants. This could be, a cow, or it could be, very importantly, this would constitute all of them. So, anything, a cow, a deer, whatever, they would eat your phytoplankton. So then from here on, we move down here to number 4, the microbial loop, very important trophic pathway. In this pathway, dissolved organic carbon is returned to higher trophic levels via its incorporation into bacterial biomass, specifically by pelagic organisms, and then coupled with the basic food chain formed by phytoplankton and zooplankton and nectin. So this is that was a mouthful. We can just summarize this by saying this is essentially how the carbon is incorporated into different organisms as they eat one another and die off, and then those nutrients get taken up by other microbes and so on. So it forms this little cycle. So essentially going back to my terrestrial environment. So this is where your deer or your antelope would then be eaten by a lion. So, and then, or in a, ocean, a macro organism example from the ocean, this is where your small fish gets eaten by a bigger fish and by a bigger fish and by a bigger fish. So this is essentially how that organic matter will then start cycling between these organisms. Okay. Okay, and important here, you will note, I'm sure, that if one smaller microbes are just eaten by larger and larger and larger, that wouldn't make a loop. So where does the loop actually come from? So the loop actually stems from the fact that, remember, these organisms can lyse, which would then release dissolved organic carbon. They can also leak some carbon. The cells can also senesce, which would release them. The zooplankton can also feed sloppily, which would mean there's some bits and pieces flying off that will also put some back in the environment. And there will also be the excretion of waste products. The waste of one organism can oftentimes be reused by another organism. So that's what actually closes our loop. So bacteria in this microbial loop actually decomposes particulate matter, and then they use this energy, during growth. So then we're going to move on to what is known as the MCP, or the microbial carbon pump. So this is essentially just the transfer of labile organic matter into recalcitrant organic matter by microbial activity. So essentially the microbes in this loop have now taken all the energy they can from these bonds, and this is the carbon that remains, the recalcitrant carbon that they cannot break down. And the microbial carbon pump is just the fraction of this long-term storage carbon that stems from the microbial loop, where the biological pump is it's exactly the same thing. It just skips the microbial loop. So it's also recalcitrant organic matter that cannot be utilized anymore. And then just a little last note here before we move on. Number 7, this viral shunt. So the viral shunt is a mechanism that prevents marine microbial particulate organic matter from migrating up trophic levels by recycling them into dissolved organic matter. This can be readily also taken up by other microorganisms. So it's another little cycle that it makes down there for us. Okay, so moving on. Sorry, that was a long slide. So let's look at some pretty pictures of bioluminescence. So bioluminescence is depicted here. You can see this glowing of organisms in the water. And these examples actually are from dinoflagellates. And when the water becomes disturbed, such as by someone rowing through them, the water will light up in this gorgeous blue hue. Yeah, it's something really pretty to look at, and that the microbes are actually behind these process. So essentially, these dinoflagellates will bioluminesce, and you can almost think of it as like a burglar alarm. So they can attract a secondary predator that would threaten to eat the primary predator. So whatever is eating them could be scared off by them, making this light and drawing other organisms that could potentially be harmful to their predators to the area. Another potential reason for this bioluminescence is just that it's driven by their disturbance. So changes in the environment, such as a drop in salinity, can then force the bioluminescent algae to glow, for instance. So those are some reasons or some thought behind why they actually bioluminesce. So this bioluminescence can actually form pretty interesting relationships with some higher organisms. So if you look at something like a Vibrio Fishery, they form symbiotic relationships with benthic dwelling fish. So these fish that live in, remember, especially in the deeper oceans where there's no light, will essentially start glowing for these fish, which will then provide them in attracting with the means of attracting and capturing prey. So how this happens is Vibrio Fishery is actually a quorum sensitive species. So essentially what that means is they're only active when a certain threshold limit in their population number is reached. So there's an auto-inducer in Vibrio Fishery, and when a certain extracellular concentration of that auto-inducer is reached, it'll trigger what is known as the LuxR to express the genes for luciferase to eventually start glowing. And the Vibrio Fishery also maintain a symbiotic relationship with the small Hawaiian squid, which it provides a safe environment. The squid will provide a safe environment for bacteria, while it'll receive aid in hunting at night. So it gets this little light that it can use. Okay, I thought that was pretty interesting. Let's move on to a less healthy portion of the work, I suppose you could look at it as. So let's look at the transmission of infected diseases. So typically this will deal with chemical quality as well as biological, but we're going to focus predominantly on biological water quality. If you guys are interested in water quality courses, I know WVU offer some others. We will delve more into physical and chemical water quality. So firstly, what I want you guys to realize at the start, when we start off with this, is that microbes can be filtered when they percolate into groundwater. So once water starts moving into the ground, it starts becoming more purified, because the impurities start to get filtered out of the water. So, but that being said, pathogens can still be transmitted to humans, especially from fecal matter that is in water. And waterborne disease, this is a stat according to the World Health Organization, that waterborne disease are responsible for over 2 million deaths annually. And most of these deaths are actually in children under 5 years of age, which obviously is terrible. And the sources of these pathogens, or what is also known as the etiological agent, is typically feces or urine of an infected person or animal. And diseases here can include bacterial diseases such as typhoid fever, cholera, which we have discussed a little bit, salmonellosis, and shigellosis. And then with protozoa, you can find amoebic dysentery, giardiosis, and encephalitis. With viruses, there are enteroviruses, hepatitis A, and Norwalk virus. So there are quite a few microbes that we have to be aware of when we are talking about bacterial water quality and biological water quality. So when we do water quality, how do we determine what the biological water quality is? Is there a biological risk inherent or contained within a water sample? So first, I'd like to start with a question. Would we ideally like to isolate the pathogens and work with these pathogens? So the answer is no. And the reason for that is actually, there are a few reasons, but the two predominant reasons are that these pathogens can be very, very hard to isolate and incubate. Their metabolisms can be rather tricky, and they can also have complex environmental requirements. But then also there's that inherent danger factor of working with these microbes. So if you work with a microbe that is known to cause disease, you need to have a specific facility, and there are some biohazard safety regulations that your lab must be, you can think of it as up to code. They have to have certain precautionary methods or precautionary measures, I should say, that would ensure the safety of the people who work with them, because otherwise they'll get sick from the microbe that they are studying or working with. So that leads us to using indicator organisms. So essentially, these are, you can think of indicator organisms as non-pathogenic organisms, but their presence are indicative of other pathogens or water quality related issues, such as fecal contamination. The E. coli that I work with a lot is a very, very commonly used indicator for fecal contamination. So these organisms can sort of provide you with a warning signal. Do keep in mind that even with this warning signal that they provide, it is not as the results you obtain are not as accurate as if you were to work directly with the pathogen. So there is a drawback there. And it can provide you with some key criteria where you can set up some water quality guidelines looking at indicator organisms, how much E. coli can be in a sample before it is deemed unsafe, for example. So just some possible indicator organisms. These are all deal with fecal contamination. First is total coliforms. So total coliform are defined as rod-shaped gram-negative non-spore-forming motile or non-motile bacteria that can ferment lactose with the production of acid and gas when incubated at 35 to 37 degrees Celsius. Now, on the other side, you have fecal coliforms. This will include E. coli. So first off, we'll start with fecal coliform because they are the same as coliform, but they can also grow in bile salts. And then E. coli, which is probably the most widely used indicator organism at this point for fecal contamination. These organisms can actually grow at up to 42 degrees Celsius. So that's higher than most coliforms, which is just an interesting characteristic of them. But the reason why these E. coli are so frequently used is they've shown the highest correlation to the outbreak of disease. So they are essentially thought to be more closely linked to pathogens. And then also the other reason is their popularity builds more popularity in a sense, where studies who want to do comparisons to other studies would prefer to use the same organism so that there isn't extra uncertainty regarding the use of different organisms, and which would make comparing results difficult. Okay. So here are some examples of water quality standards, regarding total bacteria, and then coliforms and fecal coliforms. And you can see, as you move up the list from drinking water to recreational, the limit does increase quite a bit. But I should mention here that these water quality standards may vary in different states. They can also vary in different countries. It is essentially up to the government of whatever region you're in, the people in charge, to set these guidelines. And sometimes it can be a little obscure where they get their decisions from, because typically, if a person is susceptible at a certain level, that should hold across, regardless of country or where they find themselves, but be that as it may. So there can be some differences. I thought I'd also just point out here that bottled water regulations are the same as those for drinking water from the tap. So technically, tap water quality should be the same as bottled water quality. I know a lot of people end up buying bottled water because they feel it's of higher quality, but based on the legal requirement, it's not legally required to be of higher quality. Okay, so let's look at something local that you guys should be aware of. This is our Morgantown drinking water treatment facility. This is where it is actually located. Over there, you can see there's the Mon River. There's the Aldi, and down Don Moss Boulevard, the Mercedes-Benz of Morgantown, and that's where you will find your wastewater, not your wastewater, your drinking water treatment facility. So looking at the water treatment steps involved in a typical purification plant. So first you will start, of course, with a raw water source. In our example here, our local example, this can be either from Coburn Creek. So if I jump back one, you can see this is Coburn Creek over there. Or it can be from the Monongahela River. From the raw water source, it will be moved to a pumping station. So here you can see the raw water being moved to a pumping station up there. And then from there, it will be taken to a mixing tank with alum. Alum is essentially a aluminium potassium sulfate, which aids in coagulation and sedimentation. So let's talk a bit about coagulation and infiltration. So particulates in raw water will be allowed to settle out, and flocculation will then remove the colloidal materials, including bacteria and viruses, by adding this alum. So essentially in colloidal chemistry, flocculation will refer to the process by which fine particulates are caused to clump together into a flock. So they're made into, you have all these small particles being grouped together into larger clumps or flocks. And then these super small particles can then be removed. This includes clay particles, which is the smallest component of soil, but they will actually tend to stay dissolved, as will microbes, until you add this aluminium. And then following that, or aluminum, as you guys say here. And then following that, you'll move on to filtration, which is passing water through a fine sand or coal, where the microorganisms will adsorb to the sand particles. And then following that, there will be another settling and sedimentation. So you can see the clarified water being allowed to sit out. I just want to point out here is, as you are essentially letting these particles settle out here, and also in the flocculation tank, you can see the solids are drained to the bottom there. Water is always taken from the top, because that would be the area where you don't have any solids more, or you have less solids, I should say, because they're allowed, the coarser and bigger solids are allowed to settle out, and that's how you end up purifying the water, or it aids in purifying the water. And this is what one of those tanks would look like, where the water is just allowed to stand, and for these other particles to fall out of suspension. So moving on from where we left off with the previous one, to our settling and sedimentation basin, then we will have the filtration happening. And this filter is often made of sand or activated carbon. This is sort of similar to a charcoal filter. I know many of you guys used those at high school level, so you should have some idea of how that works. And this step will also remove microorganisms, and even those that are small enough to get through the pores tend to get stuck somewhere along the way. And then you will move on to your chlorination or disinfection. And this is essentially so that any organisms that make it through the whole process up to that point will then be killed by the disinfection. So typically the disinfectant is chlorine, which oxidizes organic matter, making proteins and nucleic acids no longer functional, no longer viable. But it can also be done by UV light, especially if the water is in a very thin layer, so that the light can penetrate far enough. And you can also do ozone treatment. That's another form of disinfection that you can use. So following that, you typically have a period of fluoridation, and then you have distribution back to the consumer. So just to give you an idea of the average water use in America, so the average American family actually uses about 300 gallons per day, and this is what that split looks like. Each individual person typically uses anywhere between 80 to 100 gallons per day, and most of this is actually used to flush toilets and take showers and baths. So you can see that the shower and the toilet takes up a big proportion of our pie chart here. Here I've just included a few things and tips of ways to save water. It's worth mentioning here that the primary use of fresh water is definitely not urban environments. The main culprit, as I think many of you might be aware, is actually agriculture. So saving water in the agricultural sector would typically lead to much larger savings. Be that as it may, I think it's important for all of us to try and do our part. So, feel free to read through this list, and there are some things that would save some water and obviously save you some costs, because you wouldn't spend as much money on water every month. Okay, so now moving on to the other side of this coin. So after we've used the water and we, let it go down the drain, where does it go? So in our example, we're also going to be looking at the Morgantown wastewater treatment plant. This is what it looks like. This is sort of a picture taken from that bridge if you are on your way from the Coliseum up to Target Mountain. This is where that facility is located on the left-hand side as you are driving towards Walmart and Target. So wastewater treatment, there's a very important concept that you guys have to be aware of when it comes to sewage treatment and in general, just the ecology of waste management. And that concept is biological oxygen demand. So what that is, it is very simply just a measure of the biologically degradable organic matter in the water. So technically, it measures both the microbes and the amount of food that is present in the water, and is generally quantified as the amount of organic, it generally quantifies the amount of organic material in the water. So then also, once the water is treated, there are certain guidelines for areas downstream of the plant, where certain recreational water quality guidelines are in place to ensure that this biological oxygen demand doesn't remain very high. So primary treatment of wastewater actually ends up removing 25 to 35% of BOD. And I always thought it's important to include this last note because this helped me to conceptualize what this actually means. So essentially, your biological oxygen demand is determined by the amount of oxygen that is required by bacteria to metabolize the organic matter in the water. That's why this will include both the microbes and the amount of food or other carbon sources that is in the water. Because the more of those there are, the more of a demand on the oxygen there will be, hence the name biochemical oxygen demand. So this is that primary treatment that I was talking about, and this is essentially just the typical stages in a sewage treatment plant. Important to note here that microbial activity occurs aerobically in trickling filters or activated sludge or aeration tanks, and anaerobically in the anaerobic sludge digester. Just an overview of this picture. I do want to note out that a typical system would use either activated sludge aeration tanks or trickling filters, not both, as is shown in this picture. You typically have one or the other. You don't have both. And then also I want you guys to know that methane is produced by sludge digestion, and this is burned off or used as power, used to power heaters or pump motors. Okay, so now, as we've done so many times before, we will view and review each of these steps individually. So moving on to the primary treatment. You can think of this as like a physical process where you do some screening and grit removal and sedimentation and clarification, and this will result in a clarified liquid plus the solids, the sludge that is essentially stuck then at the bottom after they're sorted out or settled out, sorry. And this is the first stage of water treatment, where you are essentially just allowing these large solids to just settle out. And then water goes off the top to the secondary treatment. Again, similar to in the previous example, where once these solids have settled out, you will take water from the top and move that to the next secondary treatment. And then these solids are then collected off the bottom of the clarifiers, and then these go to digester tanks. Important to note that this process is pretty effective because it removes about 40 to 60% of suspended solids from the wastewater. And flocculating chemicals can increase this removal even more, and can also sometimes be added during this stage. Important also to note that biological activity is not particularly important in the primary treatment. Although some digestion of sludge and dissolved organic matter can occur during long holding times, this is not the primary reason why we do this. The sludge is removed on either a continuous or intermittent basis, and the effluent, that is the liquid that's flowing out from the top, then undergoes secondary treatment. Okay, speaking of, let's move on to secondary treatment. Here we will get to the biological component of it. So here, again, important here to note the biological oxygen demand, as we want to convert organic matter that is dissolved in the water to carbon dioxide. So we actually want to remove carbon from the water. And to do this, we use microorganisms, specifically aerobic microorganisms. So where's my mouse? There we go. Here you can see an aeration tank. And you can see how the water is like broiling and then frothing almost. And that is because the treatment plant is actually actively pumping oxygen air into this tank. And the reason for that is we wanted these processes that remove the carbon from the water are aerobic processes. So water will enter this aerobic tank. And through the aeration, we're able to stimulate aerobic organisms to metabolize the carbon that is in the water and convert it into CO2 and microbial biomass. So that is how these aeration tanks will actually function. So there's another method that we can use, and that's using a trickling filter. In this method, liquid is passed through a filter. And this filter is a matrix of solid material covered with microbial biofilms. And these biofilms essentially allow the same process to happen, where this organic material will then be converted into CO2 by these aerobic organisms. So then the water can be removed to a secondary clarifier where additional settling can occur. Here you can see the water is just being held in a stagnant, almost pool. And this is the secondary clarifier that's used for settling. And then again, the water will be drained or removed from the top. This will move us to the chlorination chamber. So this is where the water will then be disinfected. Here you can see the chlorinator. So this is essentially, we are now in the disinfection and release portion of the process. So typically, as the name suggests, it's pretty simple. Chlorine is just added to kill any remaining microorganisms. And then the disinfected effluent is then returned to the water body. So usually back into the river or wherever it comes from. So something that might strike a few of you guys, where did all the sludge go? Because remember, in that first process, we drained the sludge and then I said that will be removed, but I never told you where it goes. So essentially, after anaerobic digestion is completed, large amounts of undigested sludge will still be left over. Although this is relatively stable and inert, we want to reduce its volume, and we do this by pumping it to shallow drying beds or water extracting filters. And then following that, the sludge can be used for landfill or as a soil conditioner, and this is sometimes done under the name of biosolids. So these biosolids have been used for generations by farmers and gardeners, and they actually are so handy in reducing the need for chemical fertilizers. These biosolids are applied to promote the growth of agricultural crops, fertilize gardens and parks, and even to reclaim mining sites, pretty relevant here in coal-happy West Virginia. So when applied to the crops, the application rates are usually restricted to the nutrient needs of the crop, so that there isn't an excess of nutrients, or, which would lead to further pollution problems. The plant nutrients are slowly released by these biosolids throughout the growing season, and this will enable the crop to absorb these nutrients as they grow. This efficiency lessens the likelihood of groundwater pollution by nitrogen or phosphorus, because the rate of release matches that of the uptake, the rate of nutrient uptake from the crop. Okay. Something that I also feel we should just discuss, it wasn't really clearly illustrated on that picture because that was just a basic breakdown. The primary and secondary treatment of sewage can often not remove all biologically degradable organic matter. And amounts of this organic matter that is not excessive can just simply be released into a stream without causing a serious problem. However, should there be a increase in nutrients in these streams, this can lead to additional problems, with you are now just pumping a bunch of nitrogen and phosphorus into these streams. So you're essentially just adding a bunch of fertilizer to the stream, which would lead to eutrophication. If this is the case, additional treatment may be required. So essentially, it should also be noted that if the area that you are pumping your effluent back into is a small stream or a recreational lake, you also sometimes will find the need for tertiary treatment. And tertiary treatment is just designed to essentially remove all of the biological oxygen demand and nitrogen and phosphorus. So tertiary treatment typically depends more on the physical and chemical treatments. It's not so much biological. And phosphorus is then, in this process, is precipitated out by combining with chemicals such as lime and alum and ferric chloride. Filters of fine sands and activated charcoal then remove the small particulate matter and the dissolved chemicals. Nitrogen is then converted to ammonia and discharged into the air in stripping towers. So obviously not great that we are releasing ammonia, but it is what it is. Some systems encourage the nitrifying bacteria to form volatile nitrogen gas, much better. And then finally, the purified water is then also chlorinated again. Tertiary treatment provides water that is suitable for drinking, but the process can be extremely costly. I would also like you to keep in mind that the problem here associated with pumping this, after secondary treatment, that the water still contains 50% nitrogen and 70% phosphor. The problem with this is obviously the potential harm to ecosystems, but if we can jump down here, this problem is accelerated or exacerbated by humans because we have domestic agricultural and industrial waste being added to these streams that are now already being have this contribution of nitrogen and phosphorus, and this would be obviously constitute a further increase in the pollutants and further harm to the ecosystems. Important to note that recently there's been also a push towards using constructed ecosystems for tertiary treatment. Here you can see an example of that, where ecological processes are relied on to remove or to clean the water, essentially. Okay. So just a little PSA that I thought I'd include here. Don't drinking, it's not ideal to drink bottled water. You should more ideally add a filter to your tap if you are worried. The primary reason for this is obviously the harm to the environment, because America uses 29 billion water bottles a year, and the plastic manufacturing industry uses 17 million barrels of crude oil a year. And this is interesting here. Bottled water is not safer. There are certain exceptions. If you know you are living in an area such as Flint, Michigan, with there are no water problems, yeah, drink bottled water in that instance. But there are some things that also can make the water more unsafe, and that's typically it's plastic packaging. Where 90% of tested water bottles actually tested positive for microplastics in the water. So then you're drinking a ton of microplastics. Not great. These microplastics can have a negative impact. We're so unsure about what they do in the environment and inside of us that it's probably just better to avoid them as much as we can. And then something else I'd like to touch on here, that only 1 in 6 bottles is recycled, but this is a very misleading statistic. The reason being that the recycling process in America used to be predicated on the ability of America to export its plastics to China, where it would then be recycled. Problem is, it is much cheaper to produce virgin or unused plastics than it is to recycle plastics. So manufacturers, from a economic or capitalistic viewpoint, will obviously prefer just making new plastic. Additionally, the plastic that is produced from recycled plastic is usually inferior to virgin plastics. So it's more expensive and it produces a worse product. So from an economic standpoint, it's just not viable. Recycling is not viable. the entire narrative that the use of plastic products can be done sustainably is something that it's very much misconstrued in the public, and that's purposely done by the plastics manufacturing industry, in my view at least, because individual people deciding to recycle plastic makes such a small difference, especially because now America doesn't even recycle what they're supposed to recycle. Most of what you end up putting in the recycling ends up just going to the landfill anyway, because there are either not facilities equipped to recycle the specific polymers that you are adding into your recycling, and moreover, it also doesn't make economic sense for these manufacturers to use or to produce recycled plastic. So the entire thing here of recycling being a solution, it really isn't. When you look at plastic products, it's also one of the reasons why I am very interested now in going into plastic and microplastic research. So it's really a growing concern, and I feel the government will have to start intervening by banning single-use plastics and so forth. Anyway, let me get off my soapbox here for a second. So we're not going to have a review question today. I just thought I'd just run through the exam format with you guys. So as was the case before, we will be using Lockdown Browser again. The exams will open on 8 a.m. on Monday, the 3rd of May, and the exam submission will be available until 12 p.m. So that's noon on Friday. Please don't be late. You will not be able to submit your exam after 12. So also don't start writing your exams, at 12 or just before 12. Make sure you are done with your exams by 12. Regarding the lecture exam, that'll be 100 marks and you will have 2 hours. And the lab exam will be 75 marks or points, so 100 points, 75 points. And then the lab exam will be, you'll have 90 minutes for. So for each of these, you will have more than 1 minute per point or per mark. And the question format will be very similar to the midterm exam. So the bulk of it will be multiple choice questions. There might be a few longer questions on in the lecture exam specifically, but nothing too excessive. So you will have ample time. And then I just want to remind you guys that, remember, each exam must be completed in one sitting, meaning once you start writing your lab exam, you have to finish it. It doesn't have to be back to back. You don't have to finish both in one sitting, but whichever one you start, you have to finish. And the order of completion doesn't matter. If you want to write your lab exam first, feel free to go ahead. If you want to write your lecture exam first, that's also up to your own discretion. And guys, that'll do it. So thanks for a great semester. I think it was filled with unique challenges due to the COVID, but we're here, we're through it, we did well. I'm pretty happy about what the class average marks are at the moment. I think everyone did pretty well. And then I honestly hope that you all gain something valuable from the course, even if it's, even if microbiology isn't your major, and even if whatever you gain from the course isn't even related to microbiology. So I want you guys to have a fun and safe summer, and then I will hopefully see you again at some point. Cheers, guys.